So I have with me Jordão from CZ Biohub, and he's going to talk to us about Ultrack. Let's welcome Jordão.
Hi everyone. Today I'm going to tell about the tracking software we developed originally to track the zebrafish embryos. We are working in the Royal Lab at Jordão Bragantini Biohub. And in there we are trying to understand how the embryos tissue organized. So starting from very early development, we want to see how the cells they're picking different fate, which will result in the different organs in the organism. And I think we could take a minute to stop and see like how this process is amazing, right? Because every one of us at some point we were like a single cell, and we found the cells communicate with each other and they're coordinating and they're able to become a very complex being. And even like myself, I try to organize things with much more technology, and I have trouble organizing them. So what is cell tracking? Cell tracking, you usually want to associate the cells over time and how they're moving. And the first step is detecting the cells. A lot of times we prefer to go beyond that and segment the cells. So segmentation is where in this case, is instant segmentation for more the computer vision crowd. So we detect what is the foreground, and then for every pixel in the foreground, we map them to a single cell. So it's like clustering the pixels. And then we need to associate them to the next frame for every time point in the data set. And the challenge of this problem is because the cells, they look all the same and they can divide. So different from traditional computer vision, usually the objects have different features. In this case, they're very similar or identical, especially when you're only tracking the nuclei. And they can divide, enter, or leave the field of view. And when we started working with tracking, we realized tracking is hard because segmentation is hard. If segmentation was solved, tracking would be much easier, would be like a graph matching problem. But because segmentation is not solved, it leads to mistake that leads to tracking mistake. So because of that, we proposed Ultrack, which is a new software for joint segmentation and tracking. So we obtain a multiple set of segmentation hypotheses, which could come from different algorithms or different parameters. And then using the temporal information, we try to find the best segmentation and obtaining the tracking at the same time. And one thing we want to highlight here is the other methods that did previously tracking with multiple segmentation hypotheses, but they were constrained to a fixed set of input, for example, mixture of Gaussians for the nuclei. But in our case, we want to leverage the existing algorithms for segmentation, especially the deep learning algorithms, because people put a lot of effort obtaining, annotating the data to have very accurate models, which are very hard to beat today. At the same time, microscopy is evolving, so there are several new modalities of images being acquired, large 3D data sets, we have multicolored data sets, and we have extremely large data sets. And we wanted to develop something that could work for all these scenarios. And using the Python ecosystem, we try to do that and at least support most of the array types, so we avoid having to convert the data, because this can be a huge blocker if you have terabytes of data. So if your data can be represented as an array, could be the desk zarr array, we can process it. So just a brief review how we see how segmentation works. So we have, for example, this grayscale image on the left with the raw image. We do some kind of filtering just to smooth the image. And then segmentation could be seen as a cut, splitting the pixels apart here. So here we have 3 cells. And the cut was where we have the weakest, the lowest brightness between the pixels. But other methods, they see it as more actually a clustering problem. So they're obtaining features for each pixel. You want them to be more similar within each segment, and then you just cluster them. But essentially, it's a very simplified view is a two-step process. So you have a pre-processing and then a clustering. And I think applying this framework to I think the most widely used approach in the microscopy domain is the watershed. You have the image with its intensity. We check its profile, so we can see here we have the intensity profile of this line. And on the ridges of between the maximum of the image intensity, you define the boundary of the segments. So here we would have 3 cells. But you can see the signal is somewhat noisy. So depending how much you pre-process and smooth the intensities, you get different result. So what you want to do is to smooth enough so you get rid of the non-significant dips in the intensity, but while still maintaining the boundary that splits the cell apart. But what happens is a lot of times there won't be a unique parameter for the image. For example, here on this other profile, we have 4 cells, but in between 2 cells, there's not a significant change in the brightness. So if you run a naive approach with a single smoothing parameter, you would get 3 segments, where in reality you want 4 segments that is shown here by the dashed line. And this is applicable for several other methods. For example, in electron microscopy, it's very common to have networks that predict the cell boundary. And then they use multicut, which is a graph-based clustering algorithm, to obtain the final segments. But still, you have a parameter to tune what is the trade-off of is this a real boundary or not, because the boundaries are not perfect. And the same is applicable to cell poles, which I think is one of the most widely used 2D cell segmentation methods. They estimate the flow representation that points for each pixel the nearest centroid, and then they advect this map, and they apply the clustering. So again, emphasizing that is a two-step process, right? You obtain some features that you can pre-process or not, and then you do some kind of clustering. But I think the tricky part is doing this clustering, right? If you get the clustering wrong, tracking will be much more difficult. So we have here one image. It looks like maybe it's 1 nuclei, maybe 2. It's hard to tell. For me here, it looks like it's a single nucleus. But our idea here is to have a contour representation that tells the likelihood, or at least is proportional to the probability of being a cell boundary. So in this case, we have a stronger boundary on the outer, in the outer delineation of the cell, and then we have a weaker boundary splitting it apart, telling that there might be a boundary in there, but it's less likely. But then we obtain a hierarchical representation where we have these multiple hypotheses. And then we do this for every time point. And when we look at the whole movie, we might be able to tell if it's a single or multiple cells. So in this case, when we look at the temporal context, it becomes obvious that there were actually 2 cells that got really close for some time. And then we build our problem, and we solve the optimization problem, and we distinguish what they really were. So how we do that? We exploit the relationship between binary contours and segmentation. There's this duality. If you have binary contours, you can fill each closed contour with a distinct label, and you get the final segmentation. And you can obtain the opposite. So you have segmentation mask. You look which pixel has distinct labels, and that way you obtain the binary contour back. But it gets much more interesting when you have a fuzzy contour or a multi-level contour, because a multi-level closed contour leads to a hierarchical segmentation. And how that works is that for each level in the multi-level counter, you binarize and you apply the same methodology we did before in the binary case. So for example, if we threshold at this height of the image intensities, we get this set of counters. And as we increase the threshold, we are merging segments and obtaining our hierarchy. So this is, if you're familiar with hierarchical clustering in normal data science, not images, this is kind of doing a horizontal cut in the dendrogram, which can be naive at most of the time, and because often what we want to obtain is not a simple, the ideal result is not from a simple horizontal cut. So what we do, we develop new methodology to help picking the segments in a non-horizontal cut, but using the temporal information. So let's assume we have a very simple hierarchical segmentations for each frame. We combine, we estimate a score between the segments, and then we formulate this as an optimization problem with some constraints. So we are trying to maximize, to pick the solutions that maximize the association between the segments. So that means the segments are the most consistent over time, while we have the constraints that a pixel can only belong to a single segment. So for example here, the segment AB, when it's associated with C, we have the maximum association. And when we pick them, we block AB in the CD segment. And we formulate this taking into account as well the biological constraints, so that cells can divide, but they can only divide into two, and cells can enter and leave the field of view. So to use this in practice, we can start with I think the simplest scenario. The user has fluorescence imaging. They run an algorithm to obtain the segmentations. We convert it to a foreground mask just by binarizing the input segmentation. And we estimate the cell counters by getting the counters of the segments. And from this, we run the Ultrack, and we can obtain the cell tracks associated with the input. But the input that it supports go way beyond that. So we have 3D images of the tail bud of the zebrafish here. And we, just using differential Gaussians, we can estimate the foreground. And we can have something that is a proxy to the cell counters by just inverting the image intensities, because the boundary of the nuclei will have lower intensity. And with this, we can obtain tracks for a 3D image, which very often it requires a lot of effort because the off-the-shelf model don't have very fine-tuned weights. But here, just applying traditional image processing, we can obtain very accurate tracks. This also works for membrane data, so beyond nuclei. So here, we can use the membrane data directly because they're actually the cell boundaries. And using that, out of the box, we can obtain very accurate tracking and segmentation at the same time, even detecting the cell divisions where they get very big and then divide. But what happens when we don't have good segmentation models? I think most of the people fall, at least at some point, will have this issue. And a lot of times what the ML expert will tell you is, oh, annotate more data. But we don't want to do that, especially if we're going to 3D. So what we have here is an example where we have a few cells we want to segment. You can see some of them have very bright nuclei, but not all of them. So when we apply the cell pose, we are trying to tweak the gamma to get better contrast, but we could never reach a point where we had all the cells correctly segmented. So first we tried a very low gamma, so we were just getting the intensities, taking the power to gamma. And we would get most of the cells right, but would be a few mistakes highlighted by the red arrows. And as we tweak the parameters, we would get we would get different results, sometimes better, sometimes worse, but we would always have a little bit of mistake in the different areas. So what we can do using the framework we just developed is that we can combine all these, the contours of all these labels, obtain a fuzzy contour, and with this we can use the temporal information to do the tracking. And then looking at how the segments are consistent over time, we can obtain a result that is more accurate than what we had before. So even in the case, so here we have the ground truth, we can compute the metrics. Even if we could find the best parameter, our solution is still better than if we had that one parameter selected. And in practice, you won't, very often you don't have the ground truth, so you won't be able to find this optimal parameter, and Ultrack would be useful anyway. Another problem we were facing, this one we found on Twitter, there was someone that was looking for a cell tracking method that could track multicolored data. And the problem was we didn't have a multicolor training model. And the thing is, once again, we didn't want to train our own, right? We would have to annotate at least a few thousand of cells, and that would take a very long time. So what we did, we started evaluating each channel individually and applying a grayscale deep learning model to it. So we obtained a few segmentation masks for each channel. We noticed that because each cell has a different amount of fluorescence, some of them can be quite dim, so the model was not detecting a few cells. So we just applied a standard watershed. It's less accurate, but we have much more control. We picked a very conservative threshold, and we were able to detect more cells, even though we are not as accurate on the segmentation of splitting the instances. So what we did here, we get these 6 segmentation masks. We combine into its own contour map here, and then we just run our tracking. And we are able to track even in this environment where we have a lot of several cells. And because we have the color information, we can also use it when we are associating the weights between the different frames. So even with cells that are very dynamic and moving closer to each other, we can still have very accurate tracking. But now, I think this was the original work we were developing the tracking for. In our lab, we are imaging the whole zebrafish. So this is a 4 terabyte data set, which spans about 8 hours of zebrafish development. And here you can see how the fish didn't have a very clear structure, but now it's developing the fish head on top, and you can see the fish tail being developed. And these structures, which will become the muscle of the fish. And with this approach, we are able to track all the cells in the embryo. And here we have about 21 million segmentation instances. And the tracking is accurate even at the single cell level. So here we are tracking a single cell. You can see on the top right corner the Y-X and Z-X slice. And even as the embryo grows and it gets harder to image inside the embryo, the tracking is still accurate because we take into account this uncertainty of the segmentations. But I show a lot of nice videos how we compare with the existing methods, right? So we submitted our results to the cell tracking challenge. It's an ongoing challenge for cell tracking. It's been going for 10 years. The test data is hidden, so we can have some training data, but we don't know the scores for what we are submitting, so it's hard to overfit, and that's a good thing. This C. elegans dataset, which is one of the smaller ones, it has silver truth data, where it's a collection of the previous solution, where they created a training data set. In this one, we train a neural network to estimate the cell boundaries, and with this, we were able to achieve the first place. But the other cases here, there is no training data for the segmentation. They only have the cell centroids and their association over time, and a few 2D slices. So here we tried training our network with pseudo labels. But in the end, what did best was just a traditional image processing approach, where we do as we shown before. We detect the foreground with difference of Gaussians, and then we invert the background and apply some filtering. And I think this is remarkable because with this, first we can interpret the model. We interpret what our results much easier and tweak the parameters, and you can get things done much faster, right? I've seen many times there's like, especially new intern, they want to apply AI, and something that could take like 1 week, it takes the whole internship, and they're not finished with the project. So, and if we focus in the Drosophila data set, we have a huge increase from 0.617 to beyond 0.7. This is just using image processing. So I think this highlights how tracking and segmentation, at least in this case, is more of a clustering problem than a prediction problem. And we compared how this method scales to increasingly large data sets. So here the very tiny example of the parameter optimization using the multiple inputs from cell polls, it runs in less than 30 seconds. But we have the large zebrafish example that ran in the HPC. It took a bit less than a day to run, I think about 10 hours. So this is the large zebrafish data set. But we also compared this data set by running it in a laptop, which I think is remarkable because, of course, you're acquiring like multi-terabytes data set, but you want to be able to run that in a laptop. So we got a laptop connected to with the internet cable. We were streaming data from the HPC, and we are able to run the same experiment in 5 days. It's quite a while, but when you consider the amount of time it takes just to transfer 4 terabytes of data, it's not so bad. And it shows that we can make this really accessible to the whole community. And all of this with very few lines of code. So if you want to use this in Python and visualize in Napari, you can just follow this example. And if you want to use the HPC using Slurm, here's how we do it, just using some basic job arrays. And all of this was possible with several open source projects. One of them is not open source. So for the input and being able to load the data and process it, we use Dask, Napari to open any kind of file format as long as you have a Napari plugin, and zarr and T-file. For segmentation, we use several things. Oh. Okay. For segmentation, we use Higra, which I think is the main core for hierarchical clustering we do. But we wrote Numba, CuPy code, the out-of-memory storage of the data to be able to process in the cluster, or even large data sets in the laptop, is using SQAlchemy or SQLite and PostgreSQL. SciPy is also used, and for solving the IOPs, we use Groby, which is a commercial software, but it's open to academics. So if anyone has any recommendations for open source mixed integer programming solvers, I'm open to it. Right now I also support a Python API with Coin, but I think the development is getting slower on their side, and we are having trouble adding that to Conda. And we are able to export to several formats. NetworkX, Pandas, and even TrackMate, which is in the Java world. And I wouldn't be able to do this without my teammates in the Royer Lab at CZ BioHub and also our other collaborators. So, and thank you everyone for the attention.
Any questions?
What's your intuition on why the traditional image processing approach exceeds the AI-based approach on the cell tracking challenge? Is it sort of a lack of training data, or what's going on there?
It's a lack of training data. Most of these data sets, they are fluorescent microscopy data set where the cells were bioengineered to be easier to track. So I think that's where we have a little bit of an edge. It gets a bit easier to apply the image processing, because when you go to natural images as photographs, usually AI is much better because a lot of people spend time annotating the data, but it's harder to get a very clear boundary, for example, of a person.
Any?
Hi. Does Ultrack have support for segmenting other types of cells, like maybe like oblong cells, like myotubes?
Yes, as long as you, I think when you go to cells that are very thin, you have like a 1D segmentation problem, which can be a problem sometimes. But if you, in this case, you need a better input. If you have like omnipose or cell pose labels, it should work. But using the image intensities can be a bit more challenging, unless you have very high resolution.
Because one of the steps in your clustering algorithm was like you took the kind of the pixels and you like grouped them together. Mm-hmm. Would that work for such thin cells? Because kind of how there's the pixels can be so far apart they could like stretch the entire image.
So in these cases, I think will be a bit harder, but I don't think the distance itself is the problem, because we are looking at the image as a graph. So each pixel is connected to their adjacent ones. So as long as you have a path between the two pixels that are very far away, that should be fine. It's more that because you have like a line. If you have a single weak link there, then that can mess up your result. While when you have like a more 2D cell or even 3D, you have a lot of other ways to get to that other pixel. Does that make sense?
Yes, thank you.
Oh, it's like, yeah.
I have two questions. The first question is, when a cell divides, how does tracking happen? How do you consider the assignment of a cell to one of its daughter cells, and what happens to the other one?
Okay, so it gets a bit more complicated here.
Okay.
But the thing is, we see as a continuous flow optimization problem. So we have a slack variable for the division. You pay a price to divide, but essentially every cell needs to have the same number of incoming connections and outgoing connections, but we have some additional slack variables to make a cell appear or divide. So when a cell divide, it triggers this slack variable, and you're penalized. Otherwise, we would have unnecessary divisions, and once you pay that price, it bifurcates.
Okay, got it. And so when a new cell comes up, then it's assigned a new cell identity. Is that right? Or
yeah, kind of, because we assign the identities afterwards.
Okay.
The actual result we have is just a binary variable indicating the connections if this cell is associated to the next one.
Got it. And the other question is, what's the scaling of memory usage and compute usage with image size? I'm just kind of curious.
Okay.
And
So
the number of cells that are being tracked at the same time.
Yeah. So the segmentation, we can process, as long as you can feed a single volume and segment it in memory, you can run it. You'll be using less processes, so you'll be slower. And that's what we did in the laptop. So we had 64 gig of RAM. That was enough to segment it, but we could do only one at a time. But the problem is the IOP, because it's a combinatorial problem. It scales with, actually, I think it's polynomial scaling on the size of the number of segments we have. So what we do, we cut to be able to compute for even the large data sets. Because if we run the whole thing, we would use more than a terabyte of data, we would require more a terabyte of memory. So we process on different windows, and we have a scheduling scheme where we process first the even windows. These solutions are inserted in the database. Later they are, and when we are going to process the odd windows, we get the previous solution, we put them as constraints, so they need to be part of the solution, and then we run the problem. So this way we are able to obtain tracks that are continuous, even though we are splitting in different windows.
All right, thanks everyone for your questions. Let's thank Jordão one more time.